So, what we want to do today is continue our adventures in Chapter 5. And really, the important thing now is to stress the difference between the two kinds of respiration and then the difference between respiration and fermentation, since these are concepts that routinely get mixed up. Also, we're going to talk a little bit about the difference in these metabolic pathways when we're in a eukaryote versus a prokaryote, because they're different. And in previous courses, even if you've taken this kind of metabolic information before, it was probably taught from a eukaryotic-centric point of view. And it's interesting and useful for us to consider where things are happening and how it's working in something like bacteria, which is more relevant to this course. So the first thing we need to do is just remind ourselves what we talked about last time. It would be helpful to talk about the terminal electron acceptor and the role of NAD in all of this. So let's go to the whiteboard. So what's the first metabolic process? If we're doing respiration, which is what we're talking about right now, and we're starting with glucose, what do we do first? It's the first process that we take glucose through. If you want to metabolize glucose, what would be the first thing that you do? Break it down. What's that process called? Glycolysis. Okay. So what is the outcome of that? What do we produce? 2-pyruvate, which is a 3-carbon molecule. So we basically have chopped glucose in half. And what are the products of glycolysis besides this chemical endpoint? What are we making in the process? 2 ATPs and also 2 NADHs. Okay, so that's fairly straightforward. What are we going to do? Again, if we're doing respiration, which means we're going to get as much energy out of this as we can, where do we take our pyruvate? We first turn it into acetyl-CoA, right, which means we have to strip off one of those carbons. It gets released as carbon dioxide. And then we allow it to enter the Krebs cycle, or some people refer to it as the citric acid cycle. So we join that 2-carbon molecule with a 4-carbon molecule to make a 6-carbon molecule called citric acid. And then it goes around and it gets modified in each step by various enzymes and comes back around as a 4-carbon molecule. And how did we achieve that? We took this 6-carbon citric acid and it goes around the cycle and ends up as a 4-carbon molecule. So what happened to it? How do we get rid of those carbons? Think about this step. How did we get rid of one of our carbons from pyruvate? How do we get from pyruvate to acetyl-CoA? What is this? We're taking carbon off and releasing it as carbon dioxide. So a couple of times in the cycle, that's what happens, right? We strip off one of the carbons, releasing carbon dioxide. Now, the high energy electrons in the bond that we just chopped, where did they go? What's going to accept those high energy electrons and carry them? Right? So we put some electrons onto NAD, and we get some NADH out of this process. And there's also some ATP made in the Krebs cycle, right? So we also make a little bit of ATP directly. And what kind of way are we making ATP in that? Remember, there are 3 different ways we can make ATP. There were what? What were the names of those? There was substrate level, and then there was photophosphorylation, and then oxidative phosphorylation. So which of those 3 is happening here? And also here. Is it substrate level, oxidative, or photophosphorylation? It's not oxidative, and we know it's not photophosphorylation because photophosphorylation is when you make ATP how? By photosynthesis. So what does that leave? Substrate level. Okay, and remember, substrate level is when you basically take a phosphate group off of a molecule and stick it onto ADP, and that is substrate level phosphorylation. And we're not going through this really long intermediate, right? We're not putting the electrons first into NADH and then transporting them in the electron transport chain, then pumping protons and allowing the protons to come back and then making ATP. That whole process is oxidative phosphorylation, right? So if it's straightforward right in the reaction, it's substrate level phosphorylation. So we make some NADHs, so we make a little bit of ATP, we make a whole bunch of NADH. We also make another similar molecule, right? We take FAD and what do we make? We drop some electrons on that, FADH2. And of course, we also have carbon dioxide as waste in the Krebs cycle. So those are the first two steps. Now let's move on. What is going to happen next if we're doing respiration? So you're asking, and this is a great question, and it's a good segue. So let me just, you're sort of getting at this, right? Which is typically when you learn about respiration, this is the equation. C6H12O6, what molecule is that? What's this? This is glucose, what we're talking about up here. And if we do respiration, we turn it into what? If we burn glucose and get as much energy out of it as we can, when we, for instance, metabolize glucose, what is the final products? If we take this apart, what do we make? Right? Now these numbers are not adding up, so we'd have to do that and that. But does that sort of look familiar, right? And that's why you're asking, where is water made? This is a great question. It's not made in glycolysis, and it's not made in the Krebs cycle. But which of these is made in the Krebs cycle primarily? Yeah, so this mostly comes from the Krebs cycle. So the question is, if it's not glycolysis that's making the water, and it's not the Krebs cycle, what does that leave? What haven't we talked about yet? The electron transport chain. So, great segue, thank you for that. Let's talk about the electron transport chain. Now remember, what we've made is a little bit of ATP so far, right? And what else have we made that we're going to use now? We've been collecting all those high energy electrons. Where did we put them? NADH, right? So we have a whole bunch of NADH and FADH2, which are potential energy. They're stored, but we haven't done anything with that energy yet. So what do? Where do we take that? You take that to the electron transport chain. Here's our membrane. Remember, these electron transport chain is organized in a membrane. There's several complexes. And they drop their electrons off at the electron acceptors in the chain. And then what happens to the electron once it's accepted here? The energy from this electron then is transferred to the next, and the energy is used to do what? Pump protons, right? These are proton pumps, so they pump protons from one side to the next. Now we have a whole bunch of protons over here on this side of the membrane, whatever membrane it is. We haven't talked about that yet. So now what happens to those protons? They come back through this channel called ATP synthase. It's a channel and it's a machine. And the machine is a turbine. And as the protons come across, the energy of them coming back into the other side of the membrane is used to drive the production of ATP from ADP and inorganic phosphate that's floating around. Now, as the electrons are passed down the chain, at the end, what happens to them? Once they've run out of useful energy, who gets to take them in? The terminal electron acceptor, right? And who is that if we're doing aerobic respiration? Terminal electron acceptor, if we're doing aerobic respiration, is what? Oxygen, okay? And if we're doing anaerobic respiration, it's something else. Like what? If we're not using oxygen, what could we be using? We'd be using nitrogen, some other molecule that likes electrons. Okay, so what if we use oxygen and we put electrons onto it? That makes it negatively charged, so what is it going to be attracted to? Protons that are floating around, and that is where you make water, right? That's where the water's made. So this is just a review of what we covered last time. Did anyone have any questions about that before we sort of move on to some of the details? All right. Okay, so let's talk about the difference between aerobic and anaerobic. Now we've seen this picture, we've talked about it a lot. We have to be able to define it in words. So aerobic respiration is where at the end of the electron transport chain, the terminal electron acceptor is oxygen. If it's not oxygen, then what do we call it? Anaerobic respiration, right? So here's a question, which of these is better? Is anaerobic or aerobic respiration better, or are they equally good at making energy? What do you think? Aerobic is better, raise your hand. Anaerobic is better, raise your hand. Totally abstaining. Okay. Anaerobic respiration gives you less energy than aerobic. Now, why would that be? Can you think of a reason why that would be the case? Remember that the key difference is who's taking in the electrons at the end of the story. Okay, so why would it be the case that if you're not using oxygen but something else, you don't get as much energy? You get energy basically by transferring electrons from donor to acceptor to acceptor to acceptor. And you're going to get the most energy if you can sort of squeeze the life out of the electrons, right? So if you're going to do that, what you need is a final terminal electron acceptor that really loves electrons, even tired, ratty electrons that have no life left in them. So oxygen is that molecule. There isn't really another one in nature that's used, at least on this planet that we know of, that is as good at taking in electrons. If you use nitrogen, it's not as good. How do we know that? If you look at that electronegativity chart that you saw in the biochemistry review, then it gave you a number that tells you how good that atom is at attracting electrons. So if the number's higher, it's better. You'll see that the number for nitrogen, while similar, is a little bit lower than oxygen. So if you're going to use nitrogen or a variety of other atoms, you have to change your electron transport chain a little bit. And in doing that, it turns out you just don't get as much energy out in the end. Does that make sense to everybody? Okay. So just some ideas for what could be your electron acceptor if you're not using oxygen. Nitrate is one. If you put electrons onto nitrate, you can make nitrite, which is NO2, or nitrogen gas and water. Sulfate can also be used, and if you use sulfate as your electron acceptor, then that ends up making hydrogen sulfide gas, a toxic gas that you could smell like rotten eggs in a sulfur mine or in the volcano. You can also use carbonate, and that produces methane. So there are a variety of alternatives that are used. Are these ones that we use? Do we do anaerobic respiration? If our cells run out of oxygen, can they still respire? No. They can't. Okay, so this is stuff that microbes and other organisms on Earth do that's really important for the ecosystem or for industry, but is not something that our bodies can do. Now, can our cells survive without oxygen? What do you think? Anyone want to take the devil's advocate and say yes? So the answer is mostly no, okay? But there are some cells that can survive without oxygen, and what would allow them to do that? Can you get ATP out of any part of this process we've talked about that doesn't end up in the electron transport chain? Yes? What would you do? What's the first step in the process? What's the first metabolic pathway that we talked about? Glycolysis. So if you just did glycolysis, you could still survive theoretically. Okay, what is the downside of just doing glycolysis? There are two. Okay, what is the one downside you can think of that's the easiest to think of? Does it give you as much energy? Okay, so no, if we just do glycolysis, we're not going to get enough energy in the long run to sustain our life, right? Now, are there microbes that can live just on glycolysis? Absolutely, right? The other problem is one that we don't have a really great solution to. We have a short-term solution to, but not a long-term solution to. And I'm just going to show you really what that problem is. So let's do some glycolysis. What do we produce? Pyruvate, okay? That doesn't sound so bad. We also make a little ATP, that's also great. Now, in order to do that, we also did one other thing, which is what? When we broke this molecule, where do we put the electrons that were liberated? The reaction doesn't go without this transfer of electrons. Now, are we using this in the electron transport chain? No. So we're using this, and we're making that, but we're not going to the electron transport chain and turning it back into NAD, right? We're not dropping those electrons off so that it can come back up to the top. That's not happening. So pretty soon, we are going to run out of this. Now, what would be one way to solve that problem? We could just keep making more. That's expensive. It takes more energy to keep making more than you're going to get out of glycolysis, so long term this isn't a good solution. But in the short term, you can just run through your reserves if you just got no other options, okay? Now, is there any part of our bodies that you know of that does glycolysis that maybe can operate in the short term with like less oxygen than is optimal? Muscle, okay? So what is your muscle doing? It's going through glycolysis, and what do we know is happening with the pyruvate? What's it getting turned into? Lactic acid, okay? Now, this process is called fermentation. Fermentation is a way to deal with this NADH problem because what is happening as pyruvate is turned into lactic acid is basically that NADH is dropping its electrons off onto pyruvate, which turns it into lactic acid. That liberates NAD and it goes back to the start of glycolysis. Fermentation typically produces acids. Sometimes it also produces carbon dioxide. It just depends on the specific reaction that's happening. In this case, in our muscles, we make lactic acid. There are microbes also that make lactic acid, and that's useful in terms of making what kind of foods? What fermentation? Cheese, yogurt, right? There are actually a lot of different things that can happen here. So lots of different enzymes are involved in fermentation, depending on the specific microbial context. And that means that there are different products. So some microbes take pyruvate, make lactic acid, but some of them make carbonic acid, and some of them make propionic acid. And some of them, by the way, this is the organism that makes Swiss cheese makes propionic acid and carbon dioxide, which is why there are holes in the Swiss cheese. And there are a variety of other things that you can do to pyruvate in order to get this NADH problem solved. Just depends on what enzyme you use and specifically how you reorganize the molecule. You can have lots of different products, but most of the time it's going to be an acid. Okay? So once you've done glycolysis, if you're going to do fermentation, do you get any more energy out of this part? No. So the process, the actual fermentation part, is not an energy generating step. It's just to solve this NADH problem. That's all it is. So let's just go back to this idea. So here are some options for if your terminal electron acceptor in respiration is not oxygen. We've talked about what those things can be. But if we do use oxygen as the terminal electron acceptor, which would make this aerobic respiration, then what is our product? When we use oxygen as the terminal electron acceptor and we throw some electrons onto it, then what does it do next? It turns into water, right? It grabs some nearby protons and it turns into water. Now let's talk about where these reactions happen in the different kinds of organisms, okay? So again, if you've taken this before, then some of these answers will be easier for you, or maybe you don't remember them, which is also fine. So let's just do a little chart here. So if we are doing glycolysis in a eukaryote, where in the cell is that happening? In the cytoplasm. If we're continuing on in our eukaryotic adventure, where's the Krebs cycle happening? It's not in the cytoplasm, but many people think that it happens there, and it does happen in a fluid, which is why it's confusing. So let's skip over that for a second and talk about where the electron transport chain is located in a eukaryotic cell. Remember, what organelle is responsible for making energy? The mitochondria. So clearly the electron transport chain has to be there somewhere. Now let's just draw ourselves a little mitochondrion so that we can sort of decide where it is. Remember that mitochondria have not one, but two lipid bilayers. So here's the outer mitochondrial membrane and the inner mitochondrial membrane. This here is the intermembrane space. And then what's the center called? Does anyone remember? This is called the matrix, the mitochondrial matrix. It's a fluid much like cytoplasm, okay? And this is where the Krebs cycle takes place. And the electron transport chain is in one of these membranes. Which one do you think it is? Inner or outer? Inner, okay? So, if that's the case, let's sort of do a little diagram here. So, I'm sorry that this is going to be kind of a cluster, but here's our cell, right? Not to scale. Here's our cytoplasm. So here's our glycolysis happening, and it's making some NADH and some pyruvate, both of which have to get in to the mitochondrion. This is actually turned into acetyl-CoA, and then it goes into the matrix. That goes into the matrix also. Now we've decided our electron transport chain is here. I'm drawing it upside down just to keep this diagram from getting crazy. So our NADHs drop their electrons off here, that pass them down the line, drop them onto oxygen. We make water in the matrix, okay? Now, where are the protons going? We're pumping protons every time we transfer an electron, so where is that happening? Which direction? From what compartment to where? From the matrix into the intermembrane space. So from here, right, which is where all the stuff is happening, into this space. Now, that means there's a lot of ions, hydrogen ions, accumulating. So what does that mean about the pH of this region? It's acidic. Why is this super handy that we are putting it in this membrane between, like this space between two membranes, rather than dumping those hydrogen ions into the cytoplasm? What would happen if instead our electron transport chain was here and we were dumping protons into the cytoplasm? It would get acidic, and then what would happen? Proteins would all fall apart. Remember, we talked about the protein structure and the effect of that. So we're making water in the matrix. We're sort of driving hydrogen ion concentration up and making the pH really low in this intermembrane space. the hydrogen ions are coming back through the ATP synthase into the matrix, and ATP is also being generated in the matrix. So that's stuff that may be review for you. Let's talk about where all of this stuff happens in a prokaryotic cell. So where does glycolysis happen in a prokaryotic cell? Still in the cytoplasm. Where does the Krebs cycle happen? Still in the cytoplasm. Where is our electron transport chain? Let's draw ourselves a little prokaryotic cell. Here it is. We got some DNA, no nucleus. Here's our electron transport chain. So we've got glycolysis happening, producing some NADH and some pyruvate. In the cytoplasm, that gets turned into acetyl-CoA. That goes around the Krebs cycle. That produces more NADH. All of these NADHs go here, pass the electrons down, put them on oxygen, pump some protons outside. Why is that useful? Why is it good that we don't have our electron transport chain flipped the other way? Again, we don't want to pump protons into the cell, right? So we're pumping them outside. Now the proximal region in the outside world has more hydrogen ions, hopefully enough that we're still getting this gradient and we're allowing the hydrogens back in and generating ATP inside the cell. So this is just going to be in the slides so that you can refer to it, as opposed to my crazy drawings. Now, just to remind you of what we talked about, where fermentation is different from respiration, is that in respiration we go through all the things, right? We go through glycolysis, we go through the Krebs cycle, we go to the electron transport chain. If we're talking about anaerobic respiration, where we're not using oxygen, are we still going through all of those processes? Yeah, we are. We're just there's a little bit of a difference at the bottom. Who's collecting the electrons at the end? The specific details of the electron transport chain are a little bit different. Now, what's the difference with fermentation? We stop at pyruvic acid. We don't go through the Krebs cycle, and we don't go through the electron transport chain. Instead, we just do fermentation. We turn our pyruvate into something else, basically as a convenient way to drop the electrons that NADH has collected, and then allow glycolysis to continue. In the microbial world, different organisms have a different suite of options. We have a limited set of options. We can do aerobic respiration, and under some conditions, for short periods of time, we can do what? Fermentation, right? Producing lactic acid. But we can't do that forever. So what we cannot do of these lists is what? Which of these three things can we definitely not do? Anaerobic respiration. We do not have the ability to go through all of those things and then use something other than oxygen. That is not possible. However, there are many microbes that do. So E. coli is one fairly flexible microbe. We call this a facultative anaerobe because it's able to do both aerobic and anaerobic respiration and fermentation. Streptococcus pneumonia, which is the most common agent that causes pneumonia, doesn't have an electron transport chain. Okay, so what does that mean? No electron transport chain at all. What are its metabolic options? Can it do glycolysis? Yes, it can. But since it does glycolysis, what does it have to do next? Because it can't go through the Krebs cycle, can't go through the electron transport chain, so it stops at the bottom of glycolysis, and then what happens? Fermentation, right? So that is how it lives on sugar. It ferments. That's all it can do. So I just want to sort of alert you to the fact that you're going to run into this term fermentation in a lot of different contexts. So you may have heard of it when referring to food spoilage, right? Can you think of any examples of fermentation that are a food spoilage situation? Say again? Yeah, if milk goes sour. Now the sourness is what? What is happening? The pH is going down. It's acidic. That curdles the milk. Why? Because the proteins are unfolding. And that is a bacterial process. There are bacteria that are using the sugar in milk, which is what? What's the sugar in milk? Anyone know? A lot of people have a problem with that sugar. Lactose, right? So, but there are bacteria that are definitely not lactose intolerant. In fact, they love lactose, and they ferment. They ferment and they produce acid, which curdles the milk. Now, you also may have heard of this term in terms of making alcohol or yogurt as a fermentation process. And you may also hear it in a industrial setting. And in that setting, it often is referred to as a process that occurs without air. Is that necessarily fermentation? Is there any metabolism some microbes can do without air that is not fermentation? Anaerobic respiration, right? So there is a little bit of loosey-goosiness in the world when they use this term. When we use it in this course, we're talking about taking energy by oxidizing organic molecules in the absence of oxygen or not requiring oxygen, and definitely not using the Krebs cycle or the electron transport chain. And you do need to memorize this if it's not something that just makes sense to you. There is a terminal electron acceptor in fermentation, right? Where do those electrons go? So when we chop glucose in half, there's some electrons that get put onto NAD, turning into NADH. Where do those electrons go next? What do we do with them? How do we get NADH to turn back into NAD? We put those electrons onto pyruvate. Pyruvate then is our terminal electron acceptor. Is pyruvate an organic molecule? Yeah, why? How do we know that? It's got carbon attached to some hydrogens. Okay, so fermentation is defined as glycolysis only, no Krebs cycle or electron transport chain where the terminal electron acceptor is an organic molecule. What is the terminal electron acceptor in respiration? Is it an organic molecule? What do you think? No, it's not, right? Oxygen is not an organic molecule. Now, one of the things I told you is that in addition to nitrate and sulfate, carbonate, which has carbon in it, can also be used as a terminal electron acceptor, but it doesn't have any hydrogen, so it's not organic. Does that make sense? Now, why does this matter, besides just having memorized the definition of organic molecules? We're putting our electrons in fermentation onto a molecule that has a lot of energy still left in it. So it isn't actually a very good electron acceptor. You're only going to have that work if those electrons have not had the life squeezed out of them. There's still a lot of energy left in them, right? Whereas if we squeeze the life out of all the electrons, then we can have a terminal electron acceptor that's a low energy non-organic molecule. Okay, so we pretty much have covered all this. Where is NADH going to go in fermentation? Will NADH be used in the electron transport chain if you're doing fermentation? The answer is clearly no. What are the end products of fermentation? Sometimes you take that pyruvate and you turn it into ethanol and carbon dioxide. Now, how many carbons are there in ethanol? Anyone remember this? There's 2, right? Because if things have just one carbon, they usually are called meth something. There's 2 carbons that are called eth something, and so on. So there's only 2 carbons in ethanol. How many carbons in pyruvate? There's 3, right? We had a 6-carbon glucose. We chopped it in half. We made 2 pyruvates. So instead of making lactic acid, if you take pyruvate, you can make lactic acid, which is another 3-carbon molecule. It's just been rearranged. But if instead you drop your electrons onto pyruvate, but you have a different enzyme, and what it does is, in addition to accepting those electrons, it chops a carbon off. It makes ethanol and carbon dioxide, right? Every time you chop a carbon off, usually what you're doing is you're making carbon dioxide. Now, the part where the oxygens come in, we haven't talked about that. You don't have to know that, but just trying to help you understand where these steps are coming from. Another option is lactic acid fermentation. There are other things too. You can produce a variety of different acids. It just depends on what enzyme you're using. And some organisms are not pure in the sense that the final product of fermentation isn't just one thing, because they have a variety of enzymes that have a variety of products. Does that make sense? So they can produce, some will just make ethanol, right? So if we're using yeast, then we're just going to get ethanol, which is great because otherwise our wine would taste really weird. And if we're making yogurt, we want something that's only going to make lactic acid. But there are many things that make other products and make a variety of products. We call those mixed acid fermenters. So here's just an overview of the different things you can get. So we've got Streptococcus, Lactobacillus, and Bacillus species that when they ferment produce lactic acid. Saccharomyces, which is the yeast in beer that makes beer, wine, and bread, produces ethanol. So that's how you get fermentation. That produces like vodka or beer, and carbon dioxide, which is why, of course, when you make bread, yeast helps it to rise. That's the carbon dioxide. This is one of my favorites, propionic bacterium, which produces propionic acid, acetic acid, carbon dioxide, and hydrogen gas. So that's what helps you make Swiss cheese. Clostridium. Do we remember what genus this contains in terms of pathogens? It's the genus that contains what? 2 pathogens. These 2 pathogens make 2 of the most potent toxins. They're both anaerobic. One you get by stepping on a rusty nail, tetanus, Clostridium tetani. One you get by eating spoiled canned foods, right? Botulism. And Clostridium species have the ability to produce all of these different products when they do fermentation. You can see there's quite a range. So there's some alcohols, there's some gases, a variety of solvents. So if you want to make some solvents, you could use a bacteria. E. coli and Salmonella have the ability to make a variety of acids as well as ethanol. And Enterobacter, which is another species, or another genus that includes species that live in our intestine. All right, so I'm not going to expect you to remember the enzymatic details or even the structural details of these different kinds of fermentation, but you should understand in principle how fermentation works and that you can get different products by using different enzymes. All right, let me ask you this question. Why should we care about all of this? Why did I just drag you through all of that? What's the point besides passing this class, which is obviously the most important part? Fermentation is super important for food production, A. And also, there are pathogens that have unique signatures of fermentation ability, and we can use that to help to identify them if we're trying to diagnose somebody, figure out what's going on. If we're in research, again, having this information is really useful. You're going to do some fermentation tests, and this is what they look like in the lab. So the fermentation broth is basically very simple broth, and it will have just 1 sugar. And it varies depending on what we're testing. We'll often test, can you ferment sucrose? Can you ferment glucose? Can you ferment lactose? So we'll just put that 1 sugar in. Why is the broth red? That's because we also add a dye. That dye is pH sensitive. So the broth starts off red, so what do you think the pH is? Most likely when we're starting. It's neutral, right? Because most organisms, the ones that we're dealing with at least, have a preference like we do for a neutral pH. So this is a pH indicator that at neutral pH is red. Now, if the organism takes the sugar and does a fermentation with it instead of respiration, right, which could be either aerobic or anaerobic, but if it ferments the sugar, then what is going to happen? Usually fermentation produces what group of chemicals? Acids, which does what to the pH? pH goes down, okay? That's what's happened in these tubes here. So what does that tell you about the pH indicator? When it turns yellow, what does that mean? pH is acidic, okay? So we can do this very simple fermentation test, and we can just grow it up for a couple days, and we can see by color if it's done the fermentation process or not. Now, why is there a little glass tube in here? Besides acid, what is another possible product of fermentation? Bubbles, right? Gases. Carbon dioxide is frequently produced by fermentation. Not always. And it depends on the organism and which enzyme it's using, right? So if bubbles are formed, this tube is upside down, and it's just gonna, so if, let's say the microbe's over here and it's making gas, that gas is just gonna escape. But if it's in the tube, then the gas it makes is gonna be collected up here, and it's gonna make a bubble that you can see. This picture isn't the greatest. We will look at this in the lab. There are a variety of industrial uses for fermentation, obviously. Now, this is an important thing because it's probably one that you haven't covered in as much detail, and to be honest, we won't spend a lot of time on it either, but it is important to discuss, and that's this. If we run out of sugar, can we survive, or do we just immediately die? What else can we eat? What else do we eat? What could we live on? Protein and fats, right? The most delicious part of the food groups, in my opinion. So how do we catabolize those other organic compounds? And it's important to recognize that microbes have quite a diverse array of metabolic options, much more so than we do. But they are limited, okay? So a lot of our metabolism, obviously happens inside our bodies, but if we are, if I'm standing here and I get hungry and I'm running out of whatever's close to hand, I can walk somewhere, right? I can walk to the grocery store. I can chase down an animal and eat it, or add celery stalk if you're vegetarian, not to offend anybody. But can microbes do this? Their ability to move is limited. Now, some of them have flagella, which allows them to be motile, and some of them have an ability to sense their environment so they can chemotax towards the food. But they don't have hands, right? So they don't take the food and put it into their mouth the way that we do. What do they do instead? How do they get the food inside, right? Because the food outside in the environment is in some form that's a big molecule that can't cross the plasma membrane. And in often cases, it's even, like, let's say it's a protein, a polypeptide. Now, normally what would we do to digest that polypeptide? We put it on our mouth, we chew it up, it goes into the stomach. There are enzymes in our stomach that digest it down to the amino acids. Can microbes do that? I mean, there's the polypeptide sitting outside the cell. What do? How they get it inside? Is there some transporter that can just bring a whole protein inside the cell?
They digest it outside.
They digest it outside. How do they do that? They secrete enzymes, right? Proteases, various enzymes that will do hydrolysis reactions, right? That will just chop apart those subunits. So they secrete hydrolytic enzymes, and then once the stuff outside's been all chopped up, then those smaller pieces can be transported into the cell. So they have a variety of enzymes to do this. These are the enzymes that we would use that are inside our bodies and our organs. And apparently that's the end of the slideshow, because I think, yeah, your textbook goes into a little bit more detail about what the different metabolic pathways are for and where those go. I don't usually test heavily on that stuff. You should understand that once you've digested molecules down to their various components, that proteins, like an amino acid, can't obviously go through glycolysis. But we do metabolize glycolysis, or amino acids, in a certain way. And I'm just going to talk about that for 1 second. So if instead of glucose, where we go through glycolysis, so here's our glucose to pyruvate to the Krebs cycle to the electron transport chain. Let's suppose instead we have amino acid chain, and we hydrolyze it, and now we have amino acids. Let's remind ourselves what those look like. Right? So this here, that's a 2-carbon unit. And is there any 2-carbon step in this process that you can remember? This is 3 carbons, but before it goes into the Krebs cycle, what do we do to it? We chop off one of the carbons, making acetyl-CoA. So this would be an ideal candidate to be sort of modified a little bit to go into the into the food chain at this point if only we could get rid of some of this other crap. Okay, so one of the things we have to get rid of is the amino group. So we have to deaminate it. Remove the amino group. Now when we do that, we release this, and usually it grabs an extra proton or two. And this is ammonia, which you may know would not be a good thing to have in your body. So in our bodies, we turn it into urea. Meanwhile, the rest of this can go into, if this R group is very simple, like just a hydrogen, we can go directly into acetyl-CoA. If it's some bigger group that's got lots of carbons, then we may have to do further reactions, and we may end up feeding it into the Krebs cycle at a later step where one of those intermediates has more carbons, right? Like ketoglutaric acid or citric acid or a variety of those. You can modify this molecule depending on what it's made of and feed it into the Krebs cycle at some level. So you can even, in some cases, potentially modify it. So there are a variety of places where it can go in. It's going to bypass glycolysis, though. And now if we're doing fatty acid metabolism, let's remind ourselves what that looks like. So remember with fatty acids, let's do this sort of, look at a triglyceride, for instance. There's our triglyceride, and there's our fatty acid. These long carbon chains with and all of these have, hydrogens and whatnot. So what can we do now? What we do is this beta oxidation is what it's called. Don't really have to remember that, but we chop the chain up and release 2 carbon units that can become acetyl-CoA. So that we can take these other molecules, and basically all I need you to remember is that you feed them in just below glycolysis, somewhere in that nebulous area of the Krebs cycle, in order to keep this whole thing going, right? And we still end up making NADH, and we can still feed the electron transport chain.